Good evening to those calling in from North America. Good morning or good afternoon to people calling from around the globe. My name is Frank Wang, Head of North American Business and Development at the Shanghai Medicine. This evening, we are kicking off our Discovery Seminar Series with an excellent presentation given by Professor Zhang from University of Texas Southwestern Medical Center, which you have been all waiting for. Before Professor Zhang's talk, I would like to use a few minutes to tell who we are and what do we do. By the way, right now we have open positions, VP of Chemistry and VP of Biology at our Shanghai headquarters, and we also have BD positions at the various levels in the biotech pharma hubs across North America. You can contact me by any of those methods listed here. Medicine is a preclinical drug discovery CRO. Which is started in 2004 by Dr. Chunlin Chen, who is the current company's CEO. Over the course, we have built piece by piece of all the necessary components required for drug discovery. It has grown into a leading preclinical drug discovery development CROs in China, serving global biomedical research community. A few things I'd like to highlight here. Around 2008 and 2010, Medicine had a joint venture with a US-based CRO called MPI Research, which is subsequently acquired by Charles River. During the collaboration, our scientists were trained by all the MPI scientists, so we learned all the GLP practice, and also we inherited all those studying reports formats and the protocol formats, everything. And in 2009, Medicine was accredited by ALAC, which is the very important requirement for a company conducting animal research. And then in 2011, we got the Chinese FDA GLP certified. Also during the period, we submitted the IND packages for CFDA and the US FDA. And in 2014, we built our immuno-oncology capabilities, which including all the equipment required for doing immune profiling, such as fax machines. Also, we developed all the synthetic model and knocking model for evaluating the immuno-oncology agents. And then in 2017, US FDA had an inspection at our site, so we passed the inspection successfully. There was no issue raised. And then in 2019, our company successfully went IPO in Shanghai Stock Market Exchange. Medicine offers one-stop drug discovery services, also from target to IND. So initially from initial service, such as the targeted protein expression, structural biology, and the subsequent medicinal chemistry. And then advanced to the, once you have the lead, you like to evaluate in the tumor model or other disease models, and then also the ADME, in vitro ADME and in vivo PK capabilities, and all the way to the GLP toxicology, safety pharmacology. We also have the capability of doing the pharmaceutical development, such as API scale up and formulation formulation work. So so one thing is we certainly offer is focus the services such as chemistry or the preclinical research. We can also offer a full package for IND-enabling studies and the filing, especially data generated at the Medisalon, to support the dual filing to China NMPA and the US FDA, and the subsequently global filing. So currently at the Medisalon, we have 3 campuses all in China, and the company has about 1,600 employees. About half of them, 800 scientists are chemists. And then we have 500 preclinical research scientists. And right now we have about 800 active clients from worldwide. About one third of the top 10 big pharma right now, they have active business with us at the Medisalon. So, so as you can see, besides we have numerous regulatory IND filings to the Chinese NMPA, but we also have the regulatory IND filings to US FDA and the TGA, Australian TGA, and then the EU EMA. So now I'd like to switch the topic a little bit, say a few things about oxygen. So everyone knows oxygen is vital to life, but we probably take it for granted during our daily life. So was I before I went to a trip to Tibet in 2017 from my hometown, Boston, Massachusetts. As you know, Tibet is called the roof of the world and located in a very high altitude. It has much lower oxygen in the air. So the first evening, when I arrived in Lhasa and checked into the hotel, since there's no elevator in the hotel, I had to walk up to the 3rd floor where my room is. That was probably one of the hardest moments in my life. But after several days of acclimation, so eventually I was able to finish a marathon, in Tibet. So what happened during those several days is I traveled from sea level in Boston to like 500 meters above in Tibet. The cells in my body actually sense the low oxygen in the air, and they're reprogrammed. So I was fine in the end. So in 2019, the Nobel Prize for medicine was awarded to 3 scientists who worked on how cells sense and adapt to oxygen availability. One of them was Bill Kaelin at the Dana-Farber Cancer Institute and the Harvard Medical School. So our speaker, Professor Zhang, actually, after completing his PhD at the University of Pittsburgh Medical School, worked in Dr. Kaelin's laboratory for 7 years. First as a postdoc and then as a lecturer. He was an important contributor to Dr. Kaelin's Nobel Prize winning work. Right now, Dr. Zhang is a faculty at the UT Southwestern Medical Center, where he continued his work on the oxygen sensing pathway, but also making some groundbreaking discoveries on novel cancer therapeutic targets. So Dr. Zhang actually received many awards, one of which was a prestigious reward from American Society of Investigative Pathology, and it was announced just a week ago. So that was pretty exciting. So now let me hand over to Dr. Zhang to deliver his talk on identification and the validation of novel therapeutic targets in breast and kidney cancer.
Thank you, Frank. Thank you, everybody, for tuning in. Can you guys see my screen? Okay, so it's really my privilege to be invited by Medisalon to give this talk on some of the newer research in my lab. So some people always told me that running a lab is like running a company. The only difference is that Medisalon 16 years ago, grew from 1 person to right now about 1,600 people. For me, I started my PhD about 19 years ago. So now my lab is still fewer than 10 people. So suffice to say that medicinal really makes a lot of headway in terms of drug discovery and then those a line of work. So today I'm going to talk about the identification and validation of the new therapeutic target in breast and kidney cancer. By the way, if you guys have any questions, please type your question in Q&A session, and then I'll try to answer all of those questions at the end. So first of all, before we go into depths of the discovery research, I want to introduce a little bit about the vascular network between normal versus tumor. On the left, you can see that, I'm going to use laser pointer. This is normal tissues. You can see schematically, this is well organized, where the vasculature network is close to the normal organs, provides essential nutrients to those organs to be functional correctly. On the other hand, on the right, you can see tumor. So again, schematically, it's very tortuous, and also there's a lot of turns and twists. Also, there's some of the temporary occlusion, there's a breakage in the vessel wall, and then also there's some structure abnormalities, which as a result will cause the hypoxia to develop to distance from the blood vessels. Hypoxia also can be so-called lack of oxygen. So you would think that lack of oxygen will normally kill those healthy organs or cancer cells, but what actually doesn't kill them make those cancer cells stronger. So hypoxia is characteristic of most solid tumors. And then if you look at the clinical patient samples, I'll give you a couple of examples here. On the top, this is normal breast on the left, and then on the right, this is breast tumors. The x-axis is oxygen tensions, and the y-axis is the frequency of those patients displays oxygen tension. So if you look at the green versus red, suffice to say that it's a very dramatic shift towards left. So which means that in breast tumor, there's sort of oxygen tension shift towards lower oxygen when you compare with those normal breast tissues. On the other hand, cervix also the same deal. You can see that there's a dramatic shift to the left, showing the lack of oxygen tension in those tumors. Quantitatively speaking, under physiological conditions, most of the tissues display some kind of variance of oxygen tension. On the other hand, when we compare with the respective tumors, in most cases, in all of the cases, you can see that there's a dramatic decrease of oxygen tension in tumors compared with respective normal organs. So in terms of hypoxia and cancer therapy, you could think about this way. I mean, cancer cells grow from a very single cell to like a big round ball. So when they grow bigger, the inside of the tumors becomes hypoxic and necrotic. And outside, because they're closer to the blood vessels, they're oxygen enriched. So this is sort of diagram where you can see schematically speaking. However, when you actually patient coming in with chemoradiation therapy, in most cases, for example, radiation therapy will need oxygen to generate free radicals to kill those tumor cells. So they'll get rid of all those well-oxygenated cells on the peripheries close to blood vessels. Then those hypoxic cells or necrotic cells will be spared. And then those hypoxic cells will eventually metastasize to distant organs, kills cancer patients. So this is sort of one of the rationale people think that hypoxia may contribute to therapeutic resistance. And then in other cases, it could contribute to tumor suppressor mutations such as p53. So in any case, this hypoxia could be resistant toward therapy resistance. So the big question is that what's the molecular mechanism? So in this case, I want to introduce Bill Caton, Peter Radke, and Greg Samanza's pioneer work in the canonical oxygen sensing pathways. So you could think that under Normoxia condition, this master transcription factor called hypoxia-inducible factor alpha, including HIF-1 and HIF-2. In this case, I use HIF-1 as an example. So they will undergo this oxygen enzyme-dependent dioxygenase reaction. So where they will need alpha-ketoglutarate, they will need iron. Also, they will need essential enzyme, agonal 1, 2, and 3. So what this enzyme does is that they will convert protein residues by adding a hydroxyl group to those two conserved protein residues on HIF-1 alpha. So this modification is unique because they actually change those conformation of HIF-1 alpha, fit into those hydrophobic binding pocket of this Easter ligase called von Hippel-Lindau. This von Hippel-Lindau is an Easter ligase, but also is a canonical tumor suppressor, most mutated in kidney cancer, which I will get into. So in this case, this hydroxyl HIF, once it's modified, it will fit into the binding pocket of VHL Easter ligase, and then this VHL will take this protein to the proteasome for degradation. It's like taking to the garbage can for disposal, correct? On the other hand, for example, in tumors, because of lack of oxygen, and then this enzyme, because they depend on oxygen in order to be active, so they cannot trigger this hydroxylation reaction anymore, so they cannot be degraded. So therefore, they will translocate into the nucleus, they will bind with this heterodimer with orange. And then once we form a dimer, they're combined with HIF target genes containing hypoxia response elements. And then they can turn on downstream target genes important for tumorigenesis, neoangiogenesis, as well as erythropoiesis, migration, and invasion. For example, this is also one of the rationale which motivated development of angiogenesis inhibitor, VEGF inhibitor, such as bevacizumab in treating cancer patients. So this is a simple slide, but this is cultivated sort of like 10 to 15 years of Nobel Prize winning work, which culminated with last year's Nobel Prize in Physiology and Medicine, as Frank mentions. So I had the privilege to train with Bill Keating for 7 years. Well, Peter Radcliffe and Greg Smenza is a great leader in the field, sort of illustrate us the beautiful work of how the either low oxygen or tumors can sense oxygen tension, contributing to this sort of either physiological function or the disease progression. So our lab is interested in several perspectives. So first of all, we're interested in upstream signaling pathways. So in this way, we are mainly focused on proteohydroxylase, agonal 1, 2, and 3. So we published a few papers over the years, which I'll highlight one of the papers, Cancer Discovery for breast cancer. Also, we are also focused a little bit work on the VHL. Actually, a lot of our work on the VHL, which I want to talk about in kidney cancer. So in terms of breast cancer, why we're interested in breast cancer upstream, why we're interested in the kidney cancer downstream, there are a couple of reasons for that. First of all, we know that this enzyme will depend on alpha-ketoglutarate as a cofactor in order to be active. And then there's a subfamily of enzymes will depend on alpha-ketoglutarate in order to be active. And alpha-ketoglutarate is also essential intermediate for TCA cycle metabolism. So this cofactor or co-substrate is very important. So not too much known about alpha-ketoglutarate, although the recent years there's intensive amount of research on that topics, including many labs, including Craig Thompson and then other labs leading, including the Luke Cantley. So we decided, since we want to study this alpha-ketoglutarate, previously we have experience with agonal proteohydroxylase, we can expand this alpha-ketoglutarate to investigate whether this enzyme family, depending on alpha-ketoglutarate, may play an important role in cancer, for example, in breast cancer. So I'm going to talk about first, identification of important alpha-ketoglutarate dependent dioxygenase in triple-negative breast cancer. So why triple-negative breast cancer, or why alpha-ketoglutarate? First of all, why two alpha-glutarate is because this is a nice review in Molecular Cell a couple of years ago. Because this alpha-ketoglutarate, this enzyme family, they actually can trigger a variety of different enzymatic reactions in every single part of this physiological condition, from the DNA, histone, to transcription, to splicing, to translation, to protein stability. So the magic modification of this substrate do is that they can convert this H group to this hydroxyl group. So they can add hydroxyl group to DNA, they can add hydroxyl group to the RNA, they can add hydroxyl group into the proteins, and then they can add hydroxy group into splicing factors, and then eventually they can contribute to gene expression control. So it's very widely, functional in several different perspectives. On the other hand, why triple-negative breast cancer? So first of all, breast cancer is the #1, instance of cancer in the United States or in the world. Give me an example in the United States. Every year, there's about almost 280,000 breast cancer patients, which accounts for almost 30% of all cancers happening in human beings. And then, if you look at breast cancer, there's several different subtypes. There's ER/PR positive, which can be treated with estrogen antagonists. Also, there's HER2 positive, which can treat with HER2 antibody or tyrosine kinase inhibitors. And then there's triple-negative breast cancer, which does not express ER/PR or HER2. So that's why it's called triple-negative, which accounts to 15 to 20%. So this is a subtype which is more aggressive, have very poor prognosis, is highly heterogeneous, and then they can also relapse easily and metastasis distant organs. Also, they have lowest survival rates among all subtypes. So this is current therapeutic strategy for TNBC or for breast cancer. So you can see that in terms of metastatic breast cancers, despite that hormone receptor positive or HER2 positive breast cancer patients, they can survive approximately 5 years. On the other hand, triple-negative breast cancer, they can only survive about a year. In terms of therapy, while others, you can see there's endocrine therapy, there's targeted therapy against HER2, but triple-negative breast cancer, you can only deal with chemotherapy. Oftentimes, you can deal with 6 to 8 rounds of chemotherapy, which is brutal for the patients. You can have to deal with lethal dose adriamycin, sort of paclitaxel, and then you have to undergo 12 to 20 weeks treatment. So suffice to say, this is a cancer that needs urgent therapeutic targets. So we decided to form an angle from the TOG-dependent enzymes. So this is borrowed from my old boss Bill Cadence reviewing genes and development a few years back. There's about 70 enzymes in this family. So when we look at this one, we have a very simple mind. We'll say, let's perform a loss of function screening because there's only 70 of them. So this is to do individual one by one. In the lab, who actually was working in the industry in China for a couple of years before he came to my lab for a postdoc. So what he did is very simple. So he obtains the old-fashioned siRNA library, including those RNA against those enzymes. So there are 4 individual siRNAs. He decided to make a pool for those enzymes. And then he translated those siRNA pool into the individual well of the triple-negative breast cancer cell line, such as MDMB201. And then what he did is that he looked at 2 different phenotypes. One is that he looked at cell proliferation on the 2D. The other one is he looked at soft alga growth on the 3D, which is anchored in independent fashion, which sort of predicts this tumor growth in label. So he sort of found what are kind of the enzymes which may be important for both 2D and 3D. And then after we identify those, and he can sort of deconvolute individual siRNA, and then can sort of validate with their multiple siRNA, and then later on we use shRNA, also sgRNA, for example, just to see whether this phenotype holds true in terms of breast tumor genesis. So this is sort of end results after a long, lengthy screening platform. So basically, in order to perform screening, we have to use some kind of controls. In this case, we use GAPDH, which is housekeeping gene. What does that mean? Is that if you get rid of this gene, cells will die. And then we actually look at, on the red quadrant here, this is sort of positive hits. We found a few positive hits, and then we list some of them here. So basically, we're focusing on those protein genes when we deplete it. We can cause the defect in both 2D as well as 3D. So you will see that BBX1 is one of the proteins we focus on eventually. So it's number 7. So you would think, why would we focus on this gene? For a couple of reasons. First of all, BBX1 is one of the enzymes focused on the carnitine synthesis pathways. This is the last enzyme on carnitine synthesis. There's another enzyme called TMH-LHE. There also is upstream signaling in carnitine synthesis, also scored. showing that this enzyme family is very important for the phenotype. So, we could focus on downstream signaling BBOX1 could be a little bit more relevant. And second reason is that we also validated some other targets on the top of the list. And then turns out those proteins, when depleted, can indeed sort of deplete those TNBC cell growth. On the other hand, when we look at normal breast epithelial cells, they also kill those normal breast epithelial cells, which means that if you get rid of those genes, you will kill both normal cells as well as tumor cells. So that doesn't provide therapeutic window, because if you target it, you will kill both normal tissues as well as patient tumor tissues. So we want to sort of eliminate those toxicity towards normal tissues, but focus more maybe on the target specific for tumor cells. So suffice to say that once we did that, it's become a little bit more straightforward. So we can perform siRNA, shRNA, sgRNA, but basically just want to show you one piece of data. We eliminated the gene expression using two different shRNA in multiple TNBC cell lines. We can see that cell growth was depleted when you compare with control. And M710A and HMLE are two normal breast epithelial cells. It's immortalized, it's not perfect control, but in this case, you get rid of this BBOX1. You can see that even though in some cases it's a little bit phenotype, but grossly speaking, it's actually largely normal. So there's some therapeutic window we could potentially look at. So of course, this is an enzyme. So the burning question is that, is this enzymatic activity is important for this phenotype? This experiment is very important because if enzymatic activity is important, we can actually use inhibitors to target it. If not, and then we have to use PROTAC or other strategies to degrade the protein, which is a little bit more complicated. So Zhang used a reconstitution system, so which I can run you through. So first of all, he get rid of this BBOX1 in the cells endogenously. And then he can restore the exogenous BBOX1 either by the wild type or by enzymatic death. So in this case, he provides this kind of restoration system. So in this case, the key question is that if we use this mutant version, does that rescue the phenotype? So first of all, this control cells grow well. If we deplete this BBOX1, the cells stop growing. But if you restore this wild type BBOX1, the cell growth restores. But if you use the mutant version, despite it's highly expressed, the cells stop to grow. So what does that mean? It means that the enzymatic activity of this enzyme is important. So we can also show with colony formation assay. You can restore the duration by this wild type, but not by enzymatic death. Here the same thing with soft agar. So you can see that there's a clear cut in terms of enzymatic dependence, which forms the basis of we're using inhibitors in this triple-negative breast cancer. Of course, we want to figure out the molecular mechanism. So for this group, I'm not going to focus particularly on how we identify this, but basically we did a bunch of mass spec. We found a novel protein, IP303. It's called inositol 345 triphosphorase receptor number 3. So what is this? So we indeed can identify that indeed they bind with this IP303. Turns out this is the ER protein actually can mediate this casein release from the ER to the cytoplasm, which is uptaken by the mitochondria. So, cut a long story short, we did a lot of studies, and then eventually we found molecular mechanism studies is that this BBOX1 turns out can stabilize this IP303, and IP303 can promote this casein release. This casein release, once in the mitochondria or out of the mitochondria, on one hand, they can promote mitochondrial activity. On the other hand, they can promote mTOR activation as well as promote glycolysis. So basically, you have like double advantage. You can promote mitochondrial activity. You can also promote glycolysis. In this case, adding together, the cells will gain a lot of bioenergetics. Therefore, they can promote cell proliferation. On the other hand, if you get rid of this BBOX1, and then you will decrease both glycolysis and mitochondrial activity. Therefore, the cell will undergo apoptosis and die. So, in terms of therapeutic speaking, so first of all, we did genetic knockout experiments, knockdown experiments, which we did two different versions. One is that we basically just engineered this BBOX1 knockdown cells, and orthoptically inject into the mammary fat pads, and then monitor tumor growth over time. So you can see that in those shRNA cells, those tumors fail to grow. On the other hand, we also sort of generate inducible shRNA system. We grow the tumor first, and then we feed mice with doxycycline and get rid of Bbox1. And we found that once upon doxycycline addition, those Bbox are being depleted, and cells stop to grow, and tumors stop to grow as well. So, and then we also obtained this Bbox1 inhibitor from therapeutic angles, so which is collaboration vector and annals from the Latvian Institute of Biochemistry. So basically, this is inhibitor is a derivative version of the maltonate, which I'm going to introduce in a second. So this one is often used as a healthy supplement in athletes or other people. So we did those kind of dose-dependent experiments. We also correlate with Bbox1 expression level in those tumor cells or normal cells. So you have higher Bbox1 expression, those inhibitor tends to work better. So you have fewer lower expression, those tend not to respond as well. So also it doesn't respond in those normal breast epithelial cells. So this is all looking good, but also we want to do animal experiment. We don't have enough C2N24 from our collaborative to the animal experiment. So we turn to this compounds, it's called maltonate, which actually has been used by some of the athletes, including the former tennis star Maria Sharapova, which was banned by the Tennis Association International Federation. So because they think that this drug can sort of enhance the oxygen outputs and then sort of increase the performance of athletics in the field. But there's no direct evidence supporting that, but oftentimes people use this as a supplement for their diets. And then they'll also be treated patients in Eastern Europe for the coronary heart disease to increase oxygen outputs. So this drug, I have to say, is very safe. So we can see that in this drug, we can see the dramatic decrease of tumor volume and also tumor weight as well. But more importantly, when we treat drug over 28 days, we don't see the tumor weight, the mice weight being affected, suggesting that it's relatively non-toxic. So at this time, we're still trying to optimize this Bbox1 inhibitor. We're trying to find, can we increase the potency of this drug? Can we actually do this in the xenograft models, in the PDX models, and so on and so forth. So this is what we're shooting for. So summary one, so we identified this Bbox1 potential new therapeutic targets by engaging this hyphen and mitochondria as well as mTORC1 signaling pathways affecting those bioenergetic pathways in the cells. Okay, this is what we are actually, as an example, we focus on TNBC. On the other hand, we also focus on downstream signaling, including the von Hippel-Lindau, as I mentioned earlier on. So why we want to focus on von Hippel-Lindau, I explain in a minute, but we have been doing extensive amount of research on those tumor suppressors in kidney cancer. We are showing a few different publications we have in the past couple of years, which I explain some of the research in those publications. So von Hippel-Lindau is one of those most important tumor suppressors. The reason is simple. When you look at somatic mutation outlines of the clear cell renal cell carcinoma, which accounts for 70 to 80% of all kidney cancer, for the benefit of this talk, we can consider CCRCC is equal to kidney cancer because majority of the kidney cancer is CCRCC. So there are a variety of different genomic analysis showing that VHL sit on top of the list. They could have the nonsense mutation, missense mutation, promoter hypermethylation. So this is out of 100 patients. You can see that up to 90% of those patients carry this VHL genetic abnormalities, suggesting that this is the tumor suppressor we study in kidney cancer. So how can you target kidney cancer? So the therapeutic strategy is very simple because we know that previously by Nobel winning work, VHL can target HIF for degradation pathways. You lose VHL, you accumulate HIF, you accumulate VEGF. So therapeutic speaking, you can use either bevacizumab, sunitinib, serafenib, or pazopanib, right? This target those multi-tyrosine kinase pathways And then also you can use mTOR inhibitors, including everolimus or telomerase. So those are general protein synthesis inhibitors, which is not specific for HIF. So suffice to say, kidney cancer, therapeutic strategies very limited. So recent years, there's some HIF-2 inhibitors, but of course, it's not overwhelmingly responsive. About 50% of those patients would respond. So I stole this slide from the ESMO in 2019 conference. This is large clinical trial to look at RCC cancer patient treatment. So the standard therapy is sorafenib. So they basically either placebo, 400 patients, or in 3-year or 1-year patients with over 600 patients. So they want to see, do this drug benefits? So I can show you a very perfect negative result, which is if you look at the Kaplan-Meier, which is sort of predicts how patients fare over time upon drug treatments. So those placebo of sorafenib, treatment group totally overlap. There's no difference. There's no benefit. If you look at the other overall survival, either 3-year or 1 year. Again, you don't need to look at the details. There's absolutely no benefit. So why would those drugs, even there's no benefits, still got FDA approved? So which means that your lack of other options. So this is Winston Mannheim from NCI, which is a leading expert studying VHO. He was saying that, he's saying that we are ignoring other prospective CCRCs that may be targeted in order to achieve maximum therapeutic efficacy. So what other aspect of CCR we should focus on? That's a question we're trying to answer. Can we identify additional therapeutic avenues in clear cell renal cell carcinoma regulated by von Hippel-Lindau? So the $1 million question is how? So, how do we do that? So it's always say that I want to find some therapeutic, but the bigger question is how you can do that. So this is how our Nobel Prize winning work starts to lay the foundation for our later discoveries. So as I said early on, von Hippel-Lindau is very unique Easter ligand. They have a very hydrophobic binding pocket for substrates. So only hydroxyl HIF will bind. If it's not hydroxylated, they'll sort of turn against each other. Once it's hydroxylated, they'll fit in, okay, and then they will degrade. So if you think there's other VHO substrates, they must bind in the same hydroxyl HIF binding pockets, which means that they must bind in the same pockets. Therefore, they fit. Once they fit, they will degrade. So this is kind of hypothesis. But how can we find other things that fit into that pocket? So this is a question we have been trying to figure out. So eventually we decided to go with genomic approach. So what we did is that we obtained about 17,000 cDNA bacteria stock. There's a lot of bacteria stock. So we decided to make miniature cDNA pools. So we divided those 17,000 by 24, obtained 700. So instead of many plates, we actually have 8 96-well plates. So we can actually prep those DNAs in those 96-well plates, obtain high quality DNA. So after that, we can actually use a unique in vitro transcription translation couple system. So basically, we can directly translate those DNAs in vitro into the functional proteins. So we have used reticulolysis to maintain the appropriate post-translation modification. So on the other hand, those proteins are not labeled. There's no tag. So how can we label those proteins? So we use hardcore biochemistry, which will label those proteins as certified methionine. So that way, all of the synthesized protein will be labeled with hot radio-labeled isotopes. So that way, we can obtain those in vitro translated proteins in vitro. And keep in mind, all of them are occupied. All of them are appropriately modified, also hot labeled. So that's where the fun just started. So we decided we want to look for something X that serve as a vitro substrate. So we generate the GSD vitro complex system. If you consider this sort of triangle area is the substrate binding area, and then there's the outside area. This is basically just scaffold protein. So there's difference. Here, this is perfect triangle. Here's just specific binding on the surface. So how can we differentiate between these two? It's easy because we can use synthesized hydroxyl HIF peptide in vitro. So because those hydroxyl HIF peptides can always be excessive in nature or synthesized. So they also fit into this hydrophobic binding pockets. Also, we use a volatile HIF peptide as a negative control. They don't fit, therefore they cannot compete. And then we can perform those binding experiments and then run the SDS page. So in this case, you can see that the volatile HIF peptide cannot compete with substrate X, while the hydroxyl HIF peptide will compete. Therefore, those potential substrate will fall off. So this is how we actually perform extensive amount of thousands of GLS experiments to identify those potential proteins that may be act as substrate of von Hippel-Lindau. So then once we found that, we can deconvert the pool and then do individual binding assay one by one, just to see what novel substrate might be. So one of the earlier studies we published in 2018 in Science showing this is screening format that and then another poster in Bob Kirshner's lab in Taiwan. So this is sort of like, sort of example what we saw on hot gel. So this is what pattern we want to see. Binding with volatile peptide, but not hydroxyl HIF being competed. So this is what we see. So, out of 17,000, we come up with a couple of targets. One is called ZHH2, the other one is SFMBT1. We published this earlier paper in Science in 2018, and recently we published this paper in Molecular Cell in SFMBT1. I'm not going to go through this ZHH2, but I just want to focus a little bit more on SFMBT1 because we're sort of thinking there's a therapeutic angle we can target. So what is SFMBT1? We don't know, to be honest. At that time, there's very few publications, even though it was considered to be transcription repressor, but we think that there might be something interesting there. When we look at the structure in terms of species over the course of the species revolution, from C. elegans to the Drosophila, to the mouse, to the human. So from original just one sort of family member to the multiple family member along the way, and then there's sort of multiple brain tumor domains existing in those proteins. Multiple brain tumor domain does not necessarily mean that it has anything to do with tumor, because before our study, there's nothing reported about this protein in cancer setting. So we decided to focus on this protein, and then for a simple experiment, we look at in kidney cancer cell, we restore this VHL because they don't have VHL. So you can see that in different cells, those VHL restoration cause this protein being downregulated. So showing that this VHL can regulate this protein. And then we can look at genetic approach where we deplete this protein either by straight shRNA or inducible shRNA. We look at cell proliferation or soft agar growth. So we deplete these proteins, the soft agar growth fail to proliferate. So which means that we do the quantification, this dramatic, black and white effect. So next, we actually put those mice into the kidney also topically. So and then we monitor those tumor growth over time by using bioluminescence imaging. So first we establish tumors, and then once we know that this tumor is established, and then we add doxycycline. So once the doxycycline is added, and the SFMT shRNA starts to take effect, and then over time you can see that control cells grow well, but not SFMBT1 cells. So how about in patients? What about in patients? Do those patients carry VHL mutation, display SFMBT1 protein levels? So this is our clinical example, taking place. So where we actually obtained about 11 pairs of patient samples, we did exome sequencing, and then for normal and tumor, and then we found there's different types of mutation in tumors, including missense and frame shift and splice variants. Those are normally considered to be loss of function mutation for VHL. So in this case, in tumors, you can see clearly there's upregulation with protein SFMBT1 compared with paranormal, which recapitulates what you saw with HIF-α, which is canonical oncogene in kidney cancer, suggesting indeed there's patient relevance. So, but next question is that, this is a transcription factor. We cannot target it. What's the molecular mechanism? So we decided to take sort of unbiased approach, which means that we know that it's transcription, transcription factor. So we did the ChIP-seq for SFMBT1. We also have the ChIP-seq data for HIF-α, H3K4 monomethylation, which sort of label the transcription enhancer, H3K4 trimethylation label transcription activation, as well as promoter, H3K27 acetylation labeled active transcription. So basically, the color yellow means a highly enriched. So that's all you need to pay attention. So if you look at this SFMBT1, so don't overlap much with HIF-α, so it's HIF-α independent. That explains some of HIF-α inhibitor does not work. Maybe SFMBT1 is contributing to that phenotype. On the other hand, you can see the strong overlap with HJK4 trimethylation as well as acetylation. So by combining those factors, and we can come up with about 500 genes which fit into this category. So there's still a lot of genes to target. Can we narrow this down? So we decided to do the RNA-seq data. So if this is transcription activator modulating transcription activation, we mainly want to focus on those genes can be positively regulated by SFMBT1, which is what we show here with red color. This is all positive regulated genes for RNA-seq data. Still, we have quite some genes to work with. Can we actually add into some clinical relevance? So we decided to look in TCGA database. We look at two different things. One, if those genes are oncogenes, they should be higher in tumor, lower in normal. So we decided adding this. And if they actually are oncogenes, if they are overexpressed, then their prognosis on Kaplan-Meier should be worse, which means that they don't survive as long if they're highly expressed. So we decided to combine all those factors together into one single panel. So eventually, we found this particular gene we focus on. It's called SPHK1. It's called sphingokines 1, which catalyzes the reaction of this sphingol 1 phosphate formation, which is known to be contributing to cell proliferation in other settings. The other reason is because we have an inhibitor for that. It's a kinase. So if you look at microscopic speaking, this one you can see it's strong binding peaks. HIF-α independent, there's no binding peaks. Also, trimethylation, acetylation. So it's very well validated. And then we also see tumor is higher. We look at prognosis. High is worse prognosis. If it's medium expressed, median survival. If it's lowly expressed, better survival. And then if you look at normal versus stage 1, 2, 3, and 4, with stage higher, you can have more SFMBT1 expression. And then, as I said, fortunately, we have SPHK1 inhibitors available, which we can treat the cells with SPHK1 inhibitor. So we can see this decrease soft alcohol growth. And then we can also inject into the topically into the mice, and then we can see the short-term experiment, this tumor growth also decreased. We're also showing this drug is on target to degrade this protein of interest, but not the close family members. So basically, we're showing this phenomenon that we found this SPHK1 can be targeted in clear cell renal cell carcinoma. So it's all good, but the thing is that we're still targeting sort of downstream signaling, right? So we're not necessarily directly targeting those VHL, sort of modulatory pathways. So can we find something which is more direct? So another strategy is popular in the literature or the field is called synthetic lethality. What does that mean by synthetic lethality? So you could think about cancer cells versus normal cells. Normal cells have gene A and B or wild type. A cancer cell, this gene A is either mutated or overexpressed, the gene B is wild type. So in this case, the perfect scenario, VHL is mutated in over 70–80% of kidney cancer. So it's most widely mutated genes. Can we find something that specifically target those VHL now mutated cancers? Because that way we can spare other normal tissues. Then we found TBK1. To save the time, I won't go into specific details how we found this, but suffice to say, TBK1 is important innate immunity modulator, where James Chen's research at UT Southwestern in our next door department, the TBK1 can actually trigger this IF3 phosphorylation, therefore activate interferon, type 1 interferon response in order to sort of combat DNA RNA virus. In our case, we deplete this TBK1 by sgRNA, so either in the VHL null cells or restore the VHL cells. You can clearly say that this TBK1 only targets those VHL null cells, but not touching the VHL wild type cells. We also see another cell line, similar phenomenon, very specific. So eventually we can publish the Cance discovery paper showing that those VHL wild type cells, they actually have the low TBK1 phosphorylation. But if you lose VHL or you have tumor hypoxic, you can actually superactivate this TBK1 phosphorylation, contributing to this tumorigenesis phenotype. So it's all good, but we wanted to, is this relevant in patients? We collaborate with Canc in Peking University, showing that patients, this TBK1 is hyperactivated. So it's very highly phosphorylated in TMS beta sets. So in the cancer models, we also inject those TBK1 knockdown cells into the kidney. So over time, you can appreciate that the TBK1 knockdown cells fail to back those tumor growth. We also look at ex vivo spontaneous non-metastasis, lung necropsy. So you can see that this lung in control cells, they have high luminescent imaging data intensity, but not in the TBK1 knockdown cells. So in terms of therapeutic speaking, we actually obtained this compound 1 from David Barbee's early work in cancer discovery, which is developed by the Gilead. So this is one of the more potent TBK1 inhibitors, which shows a little bit more specificity. So in this case, just want to show you an example. In the vitro null cells, which treat drug cells with compound 1, you can see the cells start to grow. In vitro wild-type cells, largely they are actually pretty mild phenotype, or there's no phenotype, suggesting that this is sort of therapeutic angle we can potentially pursue. So at this moment, we're actually developing this specific TBK1 protein to target this TBK1 in those cells. So again, TBK1 is also potentially important for therapeutic efficacy. A couple of papers published last couple of years showing that depleted TBK1 can actually enhance this immunotherapy efficacy, either in the melanoma setting or the colon cancer setting. And then, for instance, in lung cancer setting, TBK1 inhibitor in combined with CTLA-4 can also trigger this tumor volume decrease, as well as increase the CD4 and CD8 cell populations. So suggesting that TBK1 not only sort of intrinsic cancer phenotype, but also in the immunotherapy setting could be significantly utilized as well. So if we have this potent TBK1 inhibitors, it could actually benefit immunotherapy in different cancer models. So this is meant to be explored. So take-home message is vitro perspective. We think that vitro is multifaceted protein, so which actually upon its loss, not only does that contribute to HIF-α, but also contributing to hyperactivation phospho-TBK1, as well as ZHX2, which turn on NF-κB, and also SFMBT1 pathways. It's also interesting to say there's multiple papers after our publication showing that ZHX2 is potent oncogene turn on NF-κB, not only in kidney cancer, but also in myeloma setting, as well as leukemia. So showing that this protein that sort of possesses some kind of unappreciated role. So with that, I want to thank my lab members, especially some of the people who contributed to this work, acknowledged along the way, as well as many collaborators over the years. I want to especially thank my previous mentor, Bill Caden, because the original screening for vitro Subst was started in his lab. Without his support, his guidance, I wouldn't have gone so far. But also, I want to thank all of the funding resources to support this research. And last, I want to thank for your attention, and I'd be happy to answer any questions. Thank you. Frank, can you hear me?
Yeah, I can hear you. Yeah, I can hear you. I just wonder whether do we see any question pop up on the Q&A?
Let me see. Hold on.
Maybe.
Is BBOX let me read it out. Is BBOX also upregulating other solid tumors? Very good question. We have not looked at other solid tumors in an extensive fashion. Interesting enough, we actually found BBOX1 is downregulated in kidney cancer setting. So suggesting that this is sort of tissue-specific oncogene or tumor suppressor depending on the context. In one word, in kidney cancer, BBOX1 actually is downregulated. In ovarian cancer, I'm not sure because we have not looked at ovarian cancer. But if I have to make a guess, I think that ovarian cancer especially ovarian cancer, oftentimes it sort of recapitulates some of the triple negative breast cancer. I would think that at least a subset of ovarian cancer, high grades, would have a high BBOX1 overexpression. So that's a very good question.
Dr. Zhang, actually, I have one question, just a general question. After your talk, since it's really exciting to see all these novel targets pop out from the hypoxia pathway. I just want to know, what are the sort of the, current drug development landscape for the targeting the
hypoxia pathway?
Pathway. Yeah.
Yeah, very good. You have a very good insight and a very good question. So there's several different ways. So let's first, hypoxia per se. You would think that tumor cells is hypoxic if you think that you can make tumor cell less hypoxic, or called if you can re-oxygenate those tumors. Do you think that those tumors could achieve some therapeutic efficacy? Actually, quite a few people have tried that. The result is not satisfying for the unknown reasons. So basically, they actually try to sort of infuse those oxygen into those breast tumors, but the things that actually don't work that well. So on the other hand, there are some people targeting HIF signaling because it's canonical oxygen sensing pathways. First of all, for HIF-1 alpha, there's no specific HIF-1 alpha inhibitors, so that one wanted to be developed. For HIF-2 alpha, as you guys may know, there's a specific inhibitor developed for HIF-2 alpha, which is developed by Peloton Therapeutics. Now it was bought by Merck. So right now it's called MK4682. So right now it's a phase 3 clinical trials. This drug is pretty specific for HIF-2 alpha because it targets those differences between HIF-1 and HIF-2. So they only target HIF-2 but not HIF-1. So in that case, it's really utilized in the kidney cancer setting. However, on the other hand, it's only 50%, less than 50% patient with a response, despite all of them have HIF-α mutation. And also, a lot of patients develop sort of secondary resistance towards those HIF-α inhibitors. So that actually suggesting that there's many other targets we need to look at, not only just canonical HIF signaling pathways. And the third, of course, is canonical downstream HIF signaling, including VEGF, neuroangiogenesis, including bevacizumab, which is sort of pretty much a dead horse. And then, of course, right now, a lot of people use VEGF inhibitors, acceptanib, as well as pazopanib, which is combining with immunotherapy. Actually, that strategy works pretty well because when the people use this VEGF inhibitor with immunotherapy, they found those patients actually respond much better toward immunotherapy. So that's the only angle right now people looking at this combination of therapy in combination with immunotherapy. Okay. There's a couple more questions I'll try to answer. What is the rationale of screening TOG-dependent enzyme in TNBC? That's a very good question. For a couple of reasons. First of all, our lab mainly studies TOG-dependent dioxygenase pathways, and this is enzyme family which modulates a specific hydroxylation reaction in DNA, RNA proteins. So it's understudied, and also this is sort of our niche to sort of find an angle where we can target these specific enzyme families, which we can potentially develop inhibitors to target this enzyme family. Not to say that we're not actually interested in other enzyme family, but rather we want to see, can we use this strategy sort of as a proof of principle to find something which is understudied, which we found BBOX1 is very understudied. So of course, TNBC is our to-go model because of the sort of worse prognosis and also the very little therapeutic targets. That's another scenario we use TNBC. Okay, so there's the next question. For TNBC, there's a TROP2 ADC just recently approved. Did you get a chance to look at BBOX1 expression in TROP2 ADC treated TNBC tumor model? Do you think there could be a synergistic effect from combination of therapy? Very good question. I'm going to write that down because actually I don't know about that. Actually, this is one thing I like about that because actually I can learn something useful every time when I give a talk. So we haven't looked at that. Short answer is that, but we will be actually very interested in looking into this. This is Xinyan, this is a very good question. Thank you for your suggestion. Yeah. Frank, any other questions?
Yeah, I just have one technical question. Several times during your presentation, I noticed actually you did some efficacy in the orthotopic model, broad tumors in the kidney. And
right.
I just wondering whether the reason for that, whether because of in the kidney, it's more like hypoxic conditions. So in other words, if you look at the efficacy compared with orthotopic versus sub-Q, whether you do see any difference there, whether I don't know whether it's good reasons to run with orthotopic model.
Very good question. First of all, you're absolutely right. Kidney is pseudo-hypoxic, which actually one of the reasons we like to use sort of physiologically relevant model to engage our physiological relevance. The other thing is that we want to also put those molecules in the physiological relevant system, because oftentimes subcutaneous basically does not recapitulate the tumor microenvironment with interesting study. The second obvious reason is because practically speaking, right now, for example, for the reviewers, whenever you do the subcutaneous models, they always question about physiological relevance. We went through this before several times. I know that in pharmaceuticals industry, if you want to do high throughput formats, you have to deal with hundreds of mice. You cannot do the orthotopic model. But in academia media model, because we only do like 10 to 15 mice per group each time, so we were able to do that. That's actually not a practical consideration.
Yeah, medicine can do that. Medicine has, we have a very skilled technicians who can run this orthotopic model, even some of the tumors grow inside the brain. So we have,
Right. I saw you guys, actually you do a lot of orthotopic model, but also imaging as well, because that's an important part where you can monitor tumor growth. The other reason is when you do orthotopic model, and when we actually take it out, we can also look at some of the adjacent metastases, we can look at those metastases, sort of thrombosis, those metastasis model next to the kidney, for example. So that's what we want to see as well.
Yeah. Well, that's such a outstanding talk today, Dr. Zhang. I really appreciate it. I mean, certainly I can give you a round of applause. You can hear me, but you don't hear from others.
Yeah, thank you.
Yeah. Also for the audience, in order to keep this, the such high quality, talks going on, we need your support. So like if you come today, and we hope you can come next time, and then keep us going. I mean, all we need is just your support. Coming to and enjoy such a wonderful talk, and also provide you a lot of insights about your next drug discovery efforts. Great talk, and thank you so much.
Thank you. All right. I'll see you guys next time. Thank you.
Yeah,
appreciate it.
Thank you.
Bye bye.
Appreciate it. Bye bye.